So up next we have Stella Heo, and she's a second-year master's student in psychology neuroscience behavior program at McMaster University in Hamilton, Ontario. She's working under the supervision of Dr. Nicholas Bach, and she's going to present on inter-site variability in qMRI insights from a multi-site R1 investigation.
Great. Can everyone see my screen okay? Can we start?
Yes.
Great, perfect. Then we'll get started. Thank you for the introduction and the opportunity to speak on some of the intersite variability that we observed in a multi-site R1 investigation. So multi-site research can help increase a study's statistical power and ecological validity, as we can recruit participants from larger and more diverse samples across sites simultaneously. And quantitative MRI techniques have made these multi-site neuroimaging studies more feasible by providing standardizable metrics that are comparable across different sites, theoretically. But like Dr. Stikov said, this comparability and reproducibility of qMRI data does depend on hardware configurations and sequence implementations and the vendors used. And so here we wanted to present some of the intersite variability that we observed while generating whole brain R1 maps from 2-point inversion recovery MRI data. So the data we are presenting here is part of a bigger longitudinal clinical study looking at bipolar disorder, but here we only included 3 sites that collected enough scans from healthy control participants. 2 of these sites used a 3T GE scanner, while 1 used a 3T Siemens Trio TIM scanner for image acquisition. And for this presentation, we'll just label these sites based on the vendors of the scanners used for simplicity. So at each site, participants aged 16 to 45 years were recruited to complete an MRI scan. And about a year after this baseline scan, we invited them to return to the site for a second scan. And 2 years from the baseline scan, participants were invited again to return for a third and final scan. And in total, we had 61, 55, and 23 scans from each site acquired from healthy control participants without any neuropsychiatric diagnoses. So for each scan using GE and Siemens specific sequences, we acquired a T1 weighted anatomical image and a T1 weighted image with high intracortical contrast using an inversion recovery preparation pulse and an optimized inversion time. And we also collected a T1 weighted image with low T1 contrast using a 3D gradient echo sequence with no inversion preparation. Lastly, we also acquired radio frequency transmit field maps, and all of these images for each scan were processed and linearly registered to the anatomical T1 weighted image using FSL and FreeSurfer. Then we went on to find the ratio between the 2 T1 weighted images with different contrasts and got a ratio image like the one shown here that corrected for radio frequency receive field as well as some transmit field biases. And then we used this ratio image to derive T1 estimates using lookup tables created from block simulations modeling different pulse sequences used. And we could also put in the B1 plus maps into this computation to account for any residual field inhomogeneities. So then the resulting T1 estimates were then used to calculate the longitudinal relaxation rate or the R1 maps like the one shown here. So for each scan, we then calculated average R1s for 2 ROIs, the cerebral white matter and the cortical gray matter. And lastly, for methods, we also non-linearly registered all of these images to MNI 152 standard space, then repeated the whole process to derive non-linearly registered R1 maps for each scan, which we then were able to use to average per site. So now going into results, we wanted to first show how incorporating the B1 plus maps into the block simulations affected the R1 maps. So here we have the Siemens site average R1 maps as an example. And we can see that incorporating the B1 plus field maps in computation did correct for the non-uniform shading artifacts and have made the R1s a lot more homogeneous across the brain. And when we compare these average R1 maps across the sites, we do have a different color map here just to accommodate for a range of R1 values we saw across sites. But we can see that across sites without the B1 plus correction, there is a considerable site difference between the sites, especially in the white matter. And we do see that the GE sites are showing higher R1s than the Siemens site. Now, when we do incorporate the B1 plus map in our equations, the R1s from the GE sites do become more homogeneous and decrease a bit, but we still do see intersite differences in these R1 maps. So on this slide, we have the ROI R1 plots of each scan plotted against participant age at scan. So each point here represents a scan taken, and the scans from the same individual are connected by a line. And the horizontal lines here just show the mean ROI R1s for each site. And without the B1 plus biases accounted for, we can see that there are considerable intersite differences and inter-vendor differences, with GE sites in orange and green showing higher R1s in both the cerebral white matter and the gray matter compared to the Siemens site, which is shown in purple. When we do correct for the B1 plus inhomogeneities, we can see that overall there's less intersite and inter-vendor differences to some extent in both ROIs, but again, there still is considerable variability between these sites. So just to summarize our results so far, we saw that the intersite differences in R1 maps still remain even after we incorporate the B1 plus field maps in computation. And because R1s from the Siemens site most closely resembled the values we saw in past literature, we started wondering about why the GE sites may be recording higher R1 values than expected. And one hypothesis that we came up with was that different intensity scalings or gain factors may have been applied to the two T1 weighted images without our knowledge at the GE sites. So to kind of play around with this notion, we tried to globally scale the ratio values between the two T1 weighted images using site-specific scaling factors just to see if that could somehow reconcile the intersite differences that we observed. We tried a number of global scaling factors for each site, and through visual inspection and trial and error, found that these scaling factors listed here led to considerable overlap in ROI R1s across sites. So here, just as a reminder, we just have the R1 ROI plots we saw before with the B1 plus field map incorporated in our computation. So again, the site offset is present here, but when we do apply the ratio scaling, we can see that the site differences do decrease considerably, and the data points are a lot closer to one another between sites, but there is still some site differences that we observe. And when we looked at the site average R1 maps, we saw the same results. Globally scaling the ratio values throughout the site average R1 maps a lot closer to one another, but we do still see some site differences in these maps. But because the scaling factors we used were quite rough estimates at the moment, we do believe that these results seem to support the notion that there may have been some vendor-specific and site-specific scaling of the images going on in the background. So overall, we saw considerable intersite R1 variability that remain even after accounting for different sequences that were used at sites and B1 plus field inhomogeneities. And this may be due to different intensity scalings being applied to the images, but of course there are many other possible factors that could be at play here. And regardless, we do believe that these results illustrate the need for rigorous comparisons of acquisition settings, sequence implementations, and hardware configurations and such across sites when designing and conducting multi-site qMRI studies if we do not have vendor neutral sequences available. And as for future steps, we are going to try and work towards reconciling these site differences further. Recently, we had 2 participants complete a scan at all the sites, so we're hoping to look at that data further to see if we can find a more appropriate scaling factor or any other reasons for these intersite differences. And we would also really love to hear from anyone in the audience if you have any thoughts or suggestions on what we could look at in the future. I would just like to quickly acknowledge our funding sources and everyone who have worked on this project, as well as our participants. And thank you all so much for listening.
Awesome. Thanks so much, Stella. Do we have any questions?
I do have something in the chat. I think it's more a comment than a question, but I can read it. Oh, I have something else. So from Sridhar Narayanan, is saying when implementing MTR over MT-stat on GE scanner, we have specific instructions to use manual pre-scan and hold pre-scan parameters constant between MT-off and MT-on and MTT1 acquisition. The gain settings on GE are much more fine-grained and hence variable than on Siemens. I don't know if you react.
Yeah, no, thank you for that. No, definitely, I think GE sites did show more variability for sure. So I think that is something that we could also check. Thank you.
I can add a quick comment to that as well, just because I've seen the data. So sort of what we're looking at now is potentially using the noise in the background as the scaling factor. So it didn't seem like we had similar instructions in terms of holding the gain factor constant. So we're not sure if the gain factor is changing between images. It might not be. We're certainly hoping it's not. But between sites, there's a definite difference if we just look at the histograms of the noise between the high contrast T1 and the proton density weighted image. At the McMaster site, they were quite aligned, which is what we'd hope. Same gain factor was used, but they were quite separated at the other site. So yeah, it was sort of also came up with the T1 divided by T2 weighted discussion earlier. So this definitely is a problem for, T1 mapping, but also any sort of semi-quantitative metric.
Definitely. So there is also a follow-up from Sridhar saying, asking, did you compare the correlation of T1 with age within the participant scan on the different scanners? Were the correlations consistent?
So we haven't conducted any statistical analyses with the data yet, but let's see. We can go back. We do believe that for each site, we are seeing some kind of a trend. But of course, this is qualitative and not any statistical relevance. But if we do look, we are expecting for gray matter, at least for intracortical myelin, we would expect through this age range for a slight increase and then a plateau near the end of this age range. And I think at each site, we can kind of see the plots moving up and then scaling down in terms of the increase. Though, again, I think we would have to conduct proper statistical analysis to see if that really is the case.
Thank you. So I think now we can maybe start a real discussion. So if anyone else has a question, maybe they can raise their hand.